Hello, everyone. We will wait just a minute to let the participants into the room. All right, let's get started. I am Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. And I want to welcome you to the last webinar for ASCCT and ESTIV series of 2023. And this one is a special webinar. We are doing it in partnership with our colleagues at the Japan Society for Alternatives to Animal Experiments, or JSAAE. So you can see their logo up there. Just a couple of housekeeping points to keep in mind. The webinar is recorded and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenter using the Q&A feature. The toolbar should be at the bottom of your screen usually. We'll have a Q&A period after the presenter finishes their talk. You can add your questions at any time and other participants can see them and vote for them. They really want to hear the answer to them. So again, just please put your questions in the Q&A module. We have also enabled closed captions and you can turn them on or off in the toolbar according to your preference. And please just say hello and let us know where you're joining us from. We're really excited to have you here today. So first of all, I just wanted to mention that the European Society for Toxicology in Vitro is having its conference in June of next year in Prague. And so registration is open now, so I encourage you to go to their website and check that out. And our ASCCT annual meeting will be October 28th through the 30th. We moved it slightly to accommodate an overlap with CTAC North America. So 28th through the 30th of next year. And it's going to be at the Research Triangle Institute, RTI International's Conference Center in Research Triangle Park. We should be opening session proposals and CE proposals pretty soon in the new year, so keep an eye out for that. And then, sorry, let me turn it over now to Dr. Yasu Kanda, who is head of the Division of Pharmacology, NIHS in Japan, and serves as Vice President of JSAAE.
Oh, thank you very much for the kind introduction, Christy. Can you hear me?
Yep.
I'm Yasu Kanda from NIHS in Japan, and also serve as the Vice President of JSAAE. Please let me say a few words about the session. It is our great pleasure to have a first joint webinar with ASCCT and ESTIV. As you can see here, we JSAAE had a memorandum of understanding with ASCCT and ESTIV and OSAT since before the COVID-19 pandemic. For example, we had a luncheon session and provide some JSAAE hours at the previous ESTIV meeting in Barcelona, as you can see some photos. And also, of course, we are working together with Asian groups such as KSAE in Korea and TATT in China, SAE India, and so on. And please go to the next page. Thanks. So we are very pleased to be working with you to promote 3Rs and animal alternatives. So I hope you enjoyed today's session and let's keep working together. Thank you very much.
Wonderful. Thank you, Dr. Kanda. We're, suffice to say, really excited to host this webinar so that we can all learn from each other and advance the 3Rs. So thanks for helping pull us together. Let me go ahead and introduce our speaker. So today we have Professor Masaki Nishikawa. from the Department of Chemical System Engineering and the University of Tokyo. Dr. Nishikawa graduated from the University of Tokyo and received his PhD in 2008 under the supervision of Professor Sakai, studying oxygen effects on cultured hepatocytes. Then he started to work as a postdoc and later as an assistant researcher at UCLA and the VA hospital, studying stem cell engineering and regenerative medicine on the kidney in Los Angeles, California. He moved back to the University of Tokyo in 2019 and has been a lecturer and been running the Sakai Nishikawa lab together with his former supervisor, working on various projects, including today's topic, which is evaluation of hepatocyte metabolism on a novel oxygen-permeable material with low sorption characteristics. So with that, I will turn it over to Professor Nishikawa.
Hey, hello everybody. So I'm Masaki Nishikawa. Should I share the screen, Christy?
Yes.
Okay. Can you see it?
Looks great.
Okay. Thank you very much for a nice introduction. My name is Masaki from University of Tokyo. And first of all, I'd like to thank all of you, all organizers, for giving me such a great opportunity to present our work in front of everybody. Okay, so first of all, I'd like to start with a little background about drug development and preclinical research. So to get one new drug, typically we start from 10,000 candidates of drugs, and then the selection process consists of in vivo, in silico, and in vitro approaches, followed by clinical investigations. And then this whole process in general takes about 15 years and then nearly $1 billion. And so to shorten and minimize the time and cost, it is very desired to have accurate prediction method for effects and toxicities in humans, especially without the usage of animal. So in this context, we have been working on both in silico models as well as in silico models. So as for in silico numerical models, we have been working on several models, including energy metabolisms of cells, hepatic metabolisms regarding glucose and drug metabolism, and also liver diseases include liver fibrosis. And as for in vitro MPS cell culture models, our focus has been ranging from the lung to the kidney, ended up with multiple research articles listed here. But in today's webinar, we'd like to focus on the liver model, especially regarding the effect of oxygenation through the bottom surface layer where cells are cultured. Okay, so first, let me talk about the limitations of in vitro liver models. So the first half, the top half of this slide indicates the liver structure. So here is a part of liver lobule, which is a functional unit of the liver. And then if we look at the part of this hepatic lobule at higher magnification, then we can see multi-layered structures consist of hepatocytes, hepatic stellate cells, and sinusoidal cells, in other words, endothelial cells, and on top of that, Kupffer cells, immune cells. And then the bloodstream that goes on the liver sinusoidal cells can bring oxygen, nutrients, and growth factors, so on, to the hepatocytes as well as other types of cells. And on the other hand, when it comes to in vitro cell culture systems, especially conventional cell culture conditions using TCPS tissue culture plates, there are a lot of limitations, insufficient factors to represent in vivo situations, which includes material exchanges such as oxygen, nutrients, growth factors, metabolites, and also the cell-cell interactions between hepatocytes and non-parenchymal cells are quite limited under those conditions. And so today's webinar, I'd like to put emphasis on the oxygen supply to the cells. So this slide is for explanation of limitations of oxygen supply in conventional culture conditions using TCPS plate. Typically, in the conventional cell culture conditions, the oxygen supply depends on the simple diffusion from the surface of medium to the cell layers. And by applying very typical cell culture conditions, as well as the simple fixed law of diffusion, we can calculate the oxygen flux from the medium surface to the cells, as well as the oxygen consumption rate of cells, such as hepatocyte. And then it is known that the oxygen supply from
Masaki?
the medium surface. Yes? Oh, sorry. So it is known that the oxygen supply from the medium surface is not sufficient to meet the oxygen demand of cells such as hepatocytes. So we believe that this oxygen shortage could limit the function of hepatocytes. So to overcome this kind of limitations, we thought of supplying oxygen from the bottom surface where the cells are cultured by using oxygen-permeable materials such as PDMS. And again, also applying typical cell culture conditions, we can calculate the maximum oxygen flux through the bottom oxygen-permeable membrane, which could be up to 410, which is sufficient to meet the oxygen demand of cells. So we thought that this could be a solution to the limitation of oxygen supply, which is very simple, but even effective during cell attachment, which allowed us to achieve high cell density. And so this is a result of metabolome analysis to demonstrate oxygen effect. Here you can see a line which divides two regions, one extracellular region and the other intracellular region. Within intracellular region, you can see mitochondria here. And then we measured a lot of metabolites and then mapped them into those lesions, as shown here. And overall, with low oxygen supply, we found that the cells perform glycolysis. On the other hand, high oxygen supply allows cells to perform glyconeogenesis, indicating that aerobic respirations could be achieved under high oxygen supply for energy production. And this is another demonstration of oxygen effects by transcriptome analysis. So basically, we cultured hepatocytes under different oxygen tensions from 10% to 2.5%. And then under the culture of 10% oxygen tension, which is close to the situation in artery region in the liver, the cultured hepatocytes showed very similar characteristics that we could see in the hepatocytes in the actual liver around artery region. On the other hand, the hepatocytes cultured under 2.5% oxygen tension, which is close to the situation in vein, cells exhibited very close characteristics that we can see in the hepatocyte in actual vein areas in the liver. So these results indicates that the physiological liver cell phenotypes partly, if not all, could depend on oxygen flux. And so we also, so I also tried to explain about the cell-cell interactions under this condition, such under high oxygen flux conditions. As I explained before in this webinar, in vivo liver has multi-layered structures, consists of hepatocytes and other non-parenchymal cells, which is known to be very difficult to construct in vitro due to the lack of oxygen. So we thought that by applying these oxygen-permeable materials to the cell culture systems, we could achieve to construct this type of multi-layered structures. And as you can see, we successfully created a beautiful monolayer of endothelial cells on top of hepatocytes. And besides, we could also include hepatic stellate cells in between. And also these types of complete co-culture conditions led to the higher hepatocyte functions, including urea synthesis as shown here. And so this is the summary of PDM-based MPS device. As I demonstrated, this PDMS-based MPS has clear advantages, including high cell functions of hepatocytes, which was achieved by oxygen supply and also intense cell-cell interactions. However, this culture system had clear disadvantage, unfortunately, which was sorption of chemicals, especially when it comes to hydrophobic small molecules. We could observe very severe absorption of molecules into PDMS. So this is one demonstrative results that we had. So we exposed rhodamin B to PDMS as well as TCPS. And then we found that the concentration of rhodamin B in the solution dropped very quickly after a few hours if it's exposed to PDMS, whereas almost no concentration change was observed in TCPS culture systems. So according to those sorption characteristics, not only us, but the many researchers in the world thought that this type of loss of chemicals in the system could compromise the reliability of tests. So in this sense, we had a strong desire to find a new oxygen-permeable material with low sorption characteristics. And back then, we happened to have a chance to have a discussion with Mitsui Chemicals, and then they proposed a new material, PMP, polymethylpentane, which had high gas permeable ability with low sorption characteristics. But this material hadn't been tested for cell culture use. So under the collaboration with Masaki Chemicals, we performed several investigations to check the compatibility of this material with cell cultures, including oxygen permeability test and sorption test, as well as hepatocyte culture and functional evaluations. So this is the results of oxygen permeability test. So we calculated oxygen consumption rate, in other words, oxygen flux, to the cell layer based on the oxygen concentration at the cell layer. This could be calculated by using Fick's law of diffusion, as I explained before, and this is the result. So the vertical axis is oxygen consumption rate, and then we checked, we compared it between different materials. And this blue area indicates the physiological oxygen demand of rat hepatocyte in vivo. So only in the cases where we used oxygen permeable materials, the cells could use oxygen as much as they can up to the physiological levels. On the other hand, if we culture them on regular TCPS, the oxygen consumption rate was far below to the physiological levels. So we could conclude that cell culture on oxygen permeable materials can meet oxygen demand of primary hepatocytes. And then next we moved on the sorption test. First, we used model chemicals, rhodamine B. And then, so after the exposure of rhodamine B solution, we quantified the concentration of rhodamine B in supernatants. And also we observed the cross sections of materials at the bottom surface by confocal microscopy. And this is the results of concentration change. And we compared concentrations among PMP, PDMS, and TCPS. As indicated here, after 48 hours of exposure, we observed that up to 90% of rhodamine B was absorbed into PDMS, whereas other materials didn't observe almost any. And when it comes to the results of cross sections, as you can see here, only PDMS absorbed rhodamine B into the material up to 200 micrometer from the surface of the PDMS to the deeper area. So we can conclude that PMP showed almost no sorption. And then we also checked the sorption by using CYP substrates. And as you can see, some of them was absorbed into PDMS very quickly, up to almost 100% after several hours. And then when it comes to PMP, the sorption characteristics were almost comparable with TCPS. And this is another set of substrates, and the tendency was the same. PDMS captured a lot of substrates, as indicated here, whereas PMP showed very similar characteristics with TCPS. And so we moved on to the actual hepatocyte culture on those different types of materials, and then compared the cellular functions. And for that, we cultured primary hepatocytes at this density for 7 days, and then checked the albumin secretion, gene expression patterns, and also compared CYP activities. So this is the results of albumin production. The vertical axis is albumin production, and then the horizontal axis indicates the culture days. as you can see here, the black bar, so the PMP condition as indicated in black bar could maintain higher level throughout the cell culture days. And also, as you can see in those morphological pictures, PMP could maintain a complete monolayer for at least up to 7 days. And this is the results of expression patterns of genes related to drug metabolisms. So I selected some representative results, including CYP1A2, UGT1A1, and BSEP, and MRP3. So overall, we could conclude that those expression levels were enhanced by oxygen supply, especially on PMP. And so finally, we measured CIP activity by LCMS measurements. And this is also the selected results at 4 hours after substrate exposure. And the top half of this figure shows the concentration of substrates. And as you can see, those 2 substrates were absorbed into PDMS drastically, even at the 4 hours after exposure. But the other 2 materials didn't absorbed, and then the concentration didn't change. And then under this condition, we checked the cell metabolism. And then this is a measurement of metabolites by cells. And we can see that the concentration of metabolites were higher in PMP, indicating that the functional enhancement by oxygen supply. And also, we can see that the metabolites concentrations were decreased in PDMS conditions, indicating that the unreliable measurement results, probably due to the sorption of chemicals into PDMS. So based on those results, we could conclude that PMP can be a great alternative to PDMS for the testing material, for the cell culture material. So this is a summary of hepatocyte culture on PMP. So to overcome the sorption problem into PDMS, we tested new material, PMP, and then we demonstrated the following things. First, high oxygen permeability led to higher cell functions. Second, low sorption characteristics held test chemicals in medium. Third, the both characteristics above made CIP activity test more reliable. So based on those things, we could conclude that accurate evaluation of hepatocyte metabolism could be achieved on PMP cell culture devices. However, when it comes to hepatocytes, the gold standard cells available in the market is human cryopreserved hepatocytes. So we thought that probably we need to investigate the compatibility of this culture system to human cryopreserved hepatocytes. So first, we investigated the effect of direct oxygenation on human cryopreserved hepatocytes using PDMS. So first, let me explain the results of attachment. So we compared fresh rat hepatocytes and cryopreserved human hepatocytes. Please note that both cells demand more oxygen during attachment. However, so when it comes to fresh rat hepatocytes, if we perform direct oxygenation from the bottom surface, then it improved the attachment rate and also hepatic functions as shown in the previous slides. And also the morphology after initial attachments shown here. However, if we do the same thing, the direct oxygenation to cryopreserved human hepatocytes, we observed that it exacerbated the attachment rate as shown here. So we found more detached areas of surface under higher oxygen supply conditions. So based on those results, we hypothesized that the possible damaged mitochondria during cryopreservation could lead to higher production of ROS and ended up with less attachment of hepatocytes. So to investigate and to improve that hypothesis, we modified the inoculation conditions by adding reagents and by modulating oxygen supply. So regarding reagents, the purpose of adding reagents was to suppress ROS production and also inhibit apoptosis. So in that sense, we added, tested and added those different types of reagents. The first two are antioxidants, and the second two are apoptosis inhibitors. And then we added those reagents only for the initial attachments, about 12 hours. And regarding oxygen supply, we had to meet the oxygen demand for cell attachment, but at the same time, we had to suppress the excess production of ROS. So we tried a different concentration of oxygen from 20% to 5%. And also, we performed direct oxygenation through PDMS, as well as we tried the condition where we block oxygen supply from the bottom by placing plastic seats to block the oxygen supply through PDMS, which is indicated as oxygen minus conditions instead of oxygen plus conditions. And this is the representative results of cell attachment. So if we used common cell seeding conditions, which is 20% oxygen without oxygenation through the bottom surface, we observed slight detachment of cells. And if we either increased the oxygen supply or decreased oxygen supply, interestingly, we observed either more detachment or almost no attachment under less oxygen supply conditions. And at the same time, we tried various conditions, including combinations of reagents and also oxygen concentrations. And we found that the combination of apoptosis inhibitor and direct oxygen supply at 10% concentration could give us complete monolayer as indicated here. So we concluded that controlled oxygen supply and apoptosis inhibitors improved the cell attachment. And this is the results of mitochondrial activity under the same conditions. And the vertical axis indicates the mitochondrial activity normalized to 20% oxygen blocked control. And as you can see here, this is a 20% oxygen blocked control, and then 10% oxygen control. And then this is the results obtained from 10% oxygen with apoptosis inhibitor. So as you can see here, apoptosis inhibitor didn't improve mitochondrial activity at all, which was kind of unexpected considering the results that we got on initial cell attachment rate. But interestingly, if we added antioxidants concomitantly to the system together with apoptosis inhibitors, we observed significant enhancement with mitochondrial activities as indicated here. So we concluded that the concomitant use of antioxidants and apoptosis inhibitors during attachment improved mitochondrial activity. So this is a summary so far. We achieved a more reliable test for hepatocyte metabolisms by using PMP. However, when we try to apply these culture systems to human cryopreserved hepatocytes, we observed unexpected oxidative stress during initial attachment. And then we demonstrated that the short time use of antioxidants and apoptosis inhibitors improved cell attachment and functions. So combining those two approaches, we think that we could one step closer. We could be one step closer to the accurate evaluation of human hepatocyte metabolisms. And now, Mitsui Chemicals are working on the commercialization of this new culture device, oxygen-permeable device using PMP material. And I understand it's almost available, so please contact them or please let us know if you are interested in. And also, we are now trying to use this PMP material onto the on-chip perfusion device, which was developed by Kimura Lab at Tokai University. In this perfusion device, we could perfuse medium connecting two different compartments as shown here by on-chip stirrer operated by magnet. So, which means that this system doesn't require any extra tubing and extra pumping systems, making this device very easy to operate and user-friendly. So seeing is believing. So let me show you a brief video about how to use this device, which is created by Kimura Lab at Tokai University. And here's the small stirrer, and you can perfuse medium. And this device can accommodate basically any commercially available cell culture devices, such as cell discs, as well as cell culture inserts. And it is open access, so very easy to put or remove those devices. And you can put it on the base, which can apply magnetic force to perfuse medium. And then you can also observe plates under microscope very easily. Okay, so currently we are using this device to investigate gut-liver crosstalk and also the effect of oxygenation to this type of inter-organ interactions. And long story short, we are now observing a very strong effect of oxygen, especially in liver cell functions. And we are observing that we are observing highest and most physiological liver cell functions in MPS oxygen plus conditions cultured with intestinal cells. Okay, so in this webinar, I have been demonstrating the effect of oxygen. So oxygen led to higher function, and then higher function promotes more intense interactions between cells in the same organs, as well as different organs, such as gut and liver interactions. And then we believe that the link between function and oxygen supply could be the ATP production and ATP consumption, because one glucose could only create 2 ATPs under anaerobic conditions, whereas one glucose could become 36 ATP if cells can perform aerobic respirations. So we think that the oxygen supply is a fundamental requirement to simulate in vivo human responses in vitro. Okay, so this is the summary of this webinar. So we demonstrated that new oxygen-permeable material, PMP, with low sorption characteristics could achieve more reliable test for hepatocyte metabolisms. And then when it's applied, when we need to apply this system to cryopreserved cells, including human cryopreserved hepatocytes, we demonstrated that the short-term use of antioxidants and apoptosis inhibitors could help to improve the cell attachment and also cell functions, especially for mitochondrial activities. So we believe that combining those approaches, we could be a step closer to the accurate evaluation of human hepatocyte metabolisms. And then as underlying mechanisms, I'd like to emphasize that the sufficient oxygen supply is a fundamental requirement to ensure physiological responses in vitro. So lastly, but not least, I'd like to express my deep gratitude to my co-workers and also collaborators as indicated here. That's all for today. Thank you so much for listening. I'm happy to take any questions and comments.
Well, thank you very much, Masaki. That was really a lot of interesting data leading to maybe some advice for other in vitro researchers, and certainly implications for some of the in vitro testing that we do. So I will encourage people to use the Q&A to enter your questions. We have one here in the chat. I'll just go ahead and read it out since it's the only one, and we wait for others to add their questions. So the question is, in the case of perfusion MPS, do you mean the perfusion of culture medium alone is not enough to get the higher functions and that direct oxygenation is essential?
Yep. So only by perfusing medium could not satisfy the oxygen demand, for example. So to do that, we have to perfuse medium very fast. That could apply excess shear stress and so on. So we believe that we need direct oxygenation through the bottom surface.
Okay. And another question, some compliments. They say it's an excellent system regarding sufficient oxygen supply. How many CYP enzymes are measured in the combined system. I think that's the last system that you presented. Or maybe not. In addition, could you tell me the activity of phase 2 metabolism?
Okay, so it's like here. So we tested up to 6 CRP activities. Not only this system, but also here, the simple PMP secretion systems. Yep, we checked up to 6, I believe, if I remember correctly.
Okay, good. And then what about phase 2 metabolism?
Oh, phase 2 metabolisms. Okay. So that's something we are currently working on. But let me so we need further investigation to conclude. But so it's a little bit difficult to explain. But for example, here, CRP2C9. So we observed less CRP activity in oxygen plus conditions based on the concentration of metabolites. And then we found that this was because of the enhancement phase 2 metabolisms in this cultural system. So and then also based on the other articles, those enhancement of second phase 2 metabolisms regarding this CRP activity was more physiological. That's something we could also observe in vivo. So which means that probably including phase 2 metabolisms, we could realize more reliable metabolism of hepatocyte in this culture system. But that's something we need to further investigate to conclude clearly.
Okay, thank you. Another question, have you compared the effects of some hepatotoxic substances between hepatocytes cultured on PMP and TCP?
Yeah, so internally, we observed higher, for example, higher mitochondria toxicities in PMP plates and higher sensitivity of those chemicals in PMP plates compared to the conventional secretion system. And then also, I believe we measured acetaminophen toxicities under these conditions. And then due to the higher CRP activities, such as CRP2E1, we observed higher acetaminophen toxicities in PMP plates.
Interesting.
Yeah.
Are you working on putting that data into a publication?
Yes. So you mean the toxicity data?
Yeah.
So yes. So we are currently taking acetaminophen toxicity data with this system, and then we will publish. Yes. Yep.
Okay, another question for you. With the PMP plates, how does the optical clarity and thickness compare to standard plates?
So it is completely transparent and then compatible with microscope observation. And then the thickness is about 100 micrometer to ensure the enough oxygen supply. And but it's very thin, but it's very hard. So hard material, so it can hold the shape even with the thinner thickness.
Okay. Yeah, those are important features. I see we have another question coming into the chat, so I'll read it out for you. The oxygen supply rate is changed too rapidly for cells, and defense enzymes such as SOD will not work and the cells will undergo apoptosis. So I think, is it necessary to gradually increase the oxygen concentration?
So it's yes and no. So as I demonstrated, when it comes to when it comes to cryopreserved hepatocytes, probably due to the damage of the mitochondria, it is better to gradually increase the oxygen concentrations.
Mm-hmm.
But for other cell types, we haven't observed such things. So probably we can just simply put them in the high oxygen supply conditions.
Very helpful. Another question about your discussion about acetaminophen. They're wondering which cytotoxicity tests you performed.
So far, I believe it's just the staining, and then also the leak of somehow I can't think of the exact name, but the leakage of the enzymes from the cells.
Okay.
Due to toxicity. Yeah, sorry.
No worries. It's late there. All right, so they're wondering about the higher toxicity of acetaminophen. Does that mean close to the human plasma Cmax? I don't know if you can maybe open the Q&A and you can see what they're asking.
Okay.
If that helps. But
I see. So that's something we are still investigating, but one thing I can add here is that if we change oxygen concentrations from 10% to like 2.5%, which are the range of artery to the vein.
mm-hmm.
As we see in vivo, we observed a higher toxicity of cells if they are cultured in the low oxygen concentrations, which represents the central vein area, where we see site-specific toxicity of acetaminophen in vivo. So at least I'm not sure about how quantitatively we can conclude, but at least qualitatively we can reproduce the site-specific acetaminophen toxicities by changing the oxygen concentrations, like zonation effects.
Yeah. Okay. Thank you. There's a question about other cells in the liver, in addition to hepatocytes. Well, they don't say in the liver, but I assume that's what they mean. Other cells in the liver besides hepatocytes that increase in function with oxygen supply. Or have you just looked at hepatocytes?
So we haven't investigated the functions of other nonparenchymal cells intensively, but at least I can say that activation of hepatic stellate cells could be at least partly regulated by oxygen tensions as well.
Great. I have a question myself, actually. I was wondering if there are other tissues besides liver that you think this new material would be useful for in cell culture.
I see. For example, when it comes to the oxygen consumption rate, for example, brain cells and also proximal tubule cells in the kidney could be other types of cells that consume oxygen at the higher level. So we could apply this system to those cells. And also, we could culture spheroids or cell aggregates on this material so that we can sufficiently provide oxygen into the core of the aggregates.
Yeah,
so those things could be.
Those are great thought. Yeah, I was thinking similar things. So thanks for that. So I don't have any more questions. So we're almost to the end of the hour. So I think it's good timing. I just want to thank you for your presentation, for coming to talk with us so late in your day, and for preparing your presentation. And Professor Nishikawa is a really interesting work that you're doing. And then thanks to Dr. Kanda for helping to organize this webinar. Thanks to all of you for attending. And I hope you have a nice rest of your year. We'll see you all in 2024.
Yep. Thank you very much for having me. It's been a pleasure. Thank you very much.
Yeah, thank you. It was a really great talk. Thanks for the very interesting work. All right, thank you all. Take care. Bye-bye.
Thank you. Bye.